So thank you very much. I'm Bob Molinari, the founding CEO of Repoxegen Therapeutics. And just want to make sure that what I'm saying there. So to the point that was just made, you have to start somewhere. This is a platform technology, and our first indication is going to be non-proliferative diabetic retinopathy. This is somewhat of an unmet medical need. Before you have symptoms, most people don't want to have injections in the eye. And the unmet medical need is really, is there a treatment other than watchful waiting, which is sort of the standard of care right now? What we're developing is an oral drug that is disease-modifying in this disease. It can likely prevent the disease from going forward and eventually needing those injections, and we think it's a new opportunity. So why does this opportunity exist? We have a whole new point of view on the product. That is, what are we seeing there? Yeah. So we are seeing, we are targeting polyunsaturated fatty acids. These are all over in the retina, particularly DHA, arachidonic, are highly prevalent in the retina. If you look at these fats the wrong way, they degrade. And there's a whole metabolism around the degradation. Most of the things they degrade into or metabolize into are on the right. They are retinal and neuroinflammatory triggers. Things like prostaglandins, thromboxanes, products of TNF-alpha, interleukins. There's a little-known group of these that are actually degradation products of the fat that are neuroprotective. And in general, there's supposed to be a balance between those two. And you don't get what you see in the lower left here, which is unresolved inflammation. When you have the disease state of diabetic retinopathy, or for that matter, AMD, or any host of other retinopathies and even neuropathies, you see there is an enzyme called SEH, soluble epoxide hydrolase. That green area are a small class of epoxy fatty acids that are actually protective of the retina. When you have the disease, the enzyme is upregulated that converts those neuroprotective compounds into neurodegenerative ones, or retinal degenerative ones. It's kind of like a master switch. And if you actually look at human diabetic retinopathy patients and you stain for that enzyme, the soluble epoxide hydrolase, you can see that it goes up in lockstep with severity in diabetic retinopathy. And that's shown on the right as well. So what happens if you inhibit that enzyme? This is drug discovery 101. We have an enzyme, we have a small molecule that can inhibit. What happens? So in the Akita mouse model, you can see that when you have the severe disease, you strip off the capillaries in the retina, the impermeability layer, which is the VE cadherin layer. That causes the capillaries to be leaky. It can lead to extracellular capillary formation and the pathology of the disease. All you do is inhibit this enzyme with a small molecule, and you can recreate that capillary layer. And when you do, on the right, you'll see in the upper right quadrant, there's a lot of extracellular capillary formation. In the lower right quadrant, that goes away. So this is truly, via this mechanism, disease-modifying therapy. It doesn't just stop there. If you do a CNV model, generally the target of anti-VEGF drugs, they work extremely well at sort of stopping the progression or putting a finger in the dike, you might say. And the beauty of these indications are you can actually look in the eye, stain for, and see these lesions. So if you have the untreated lesions on the left and you treat them with anti-VEGF, you see them starting to decline. If you treat them with an earlier generation soluble epoxide hydrolase inhibitor, you can see them go away as well. Interestingly enough, if you actually combine the two, you can see even better efficacy. So maybe this is a way to either do monotherapy instead of these injections in the eye, that is oral, You're probably not going to start there because we don't want to do superiority over VEGF. It works pretty darn well. But maybe a combination therapy could extend the time it required to take those injections. And you can see that this is actually dose-dependent in the lower right. So how do we differentiate ourselves? Well, it's pretty easy given some of the discussion we've heard as to how you differentiate yourself in this environment. And we are oral. We are a daily pill. And it's traveling, and it's focused on this inflammatory cascade that goes on. And we're also disease-modifying in terms of reducing the size of these lesions that lead directly to blindness. So we're talking about platform companies. It works in academic studies in earlier versions of the drug in all of those things on the left in the retina: diabetic retinopathy, glaucoma, macular edema. There are studies that show that this has high disease-modifying potential. We're focused on diabetic retinopathy really because of the TPP. The product profile of an oral drug in something that's untreated now is quite attractive. By the way, the drug was optimized by a group in Barcelona that's mainly interested in neurology. There is equally good data, stunning data, in fact, in a whole bunch of neuropathies. And to be focused, we're not going there. Those are trials that they're subjective, they take very long periods of time, and they really, very large cohorts. Whereas in the eye, you can have an early readout, even if it's not what the FDA will allow you to get approved. You can get an early readout of, is the drug working, by looking at the dead cells and essentially the area of those dead cells. So where are we and what are we asking for? I guess we might be part of the valley of death discussion as well, because we are, this drug class, these soluble epoxide hydrolase inhibitors, have been studied over a decade. They have been in human as a class. They were not developable because over the years, the IP around that class really got muddled and aged out and a bunch of other things. So we didn't start this company until we had a license to a 5-year structure-activity relationship that did the hard work of turning these inhibitors into drug-like compounds. So looking at therapeutic window, solubility, dosing, all those other things. And that actually created a whole new set of IP that we were able to license in. And we are now ready to nominate a lead compound from that work for IND-enabling work. That'll take about $5 million. That's what we're initially raising. Some people in the industry would like to see a first milestone for funding all the way through to human proof of concept. With another $25 million, we expect to get into the phase 1b/2a study in the eye and be able to do that, with the major endpoint being, again, the lesion reversal. The core team here is an experienced group of drug hunters. I actually was, over the last dozen years or so, one of the pioneers of the notion that you ought to be targeting fats in the brain and the retina. The brain and the retina are largely composed of fat, some 60%. There's a joke here. If someone calls you a fathead, consider it an honorific. And we also have an experienced team. So Ving Li is a CMC specialist who worked in the area of soluble epoxide hydrolases, as well as running a CRO of his own. So he's got all the things we need to scale up and manage those kinds of contracts. Sherif Mehta, who is here as well, is an accomplished business development person who knows how to add value to these companies. He's written a book about it that's a highly regarded textbook. And Ken Mandel is probably someone known to you as well. He is coming on board right now to help us as an advisor to really fine-tune the retinal development strategy. And then, of course, as most companies of this type, we have the usual key opinion leaders. You probably recognize some of the people here: Gary Novak, Kwandan Nguyen, who are helping us with this strategy as well. So I think with that, I'll stop.
So Bob, mainly, have a seat.
Sure.
I think at this point, Riad would have come on the podium, and we would have had this discussion together. Yep. In reality, Riad was stuck in London because of weather, so you're going to have to be his voice as well. If that's okay with you.
Okay, sure.
So
Oh, it's much better. You're really a silhouette over there.
it's kind of like we'll let them sit over there once the other panel members are coming on board. So my question to you is, regarding the fact that you are very early stage, and you are still, presenting this as a potential platform with a lead potential compound. What are the challenges that you're facing in coming to speak with investors, and how, through the discussions that you've had with Riad, he was maybe able to offer you some advice on that?
So triaging on indication is key here, because we have so many that we think we can actually consider. And what Riad was helping us with is, how do you winnow down that list? How do you think about what you present? And again, with a technology product profile that's interesting enough to be a large market, competitively attractive against whatever the state of the art is. And my experience in startups is, when you do that, that is the company you become. Doesn't matter what all those other things are. And I mentioned we're in retinopathies because we really like the clinical trial dynamics there. You are able to look into the eye with OCT and see these lesions. You're able to see extracellular capillaries. Regardless of the specific pathology, that is very compelling from a pharmaceutical point of view, to know the drug is engaging the target, is working, is doing what you think. And even the FDA accepts that, dead cells usually mean eventual loss of vision. And that's what we're trying to measure, is the sort of area of those dead cells.
And so in pitching this to investors, what were some of the feedback that you've received?
So I think the investors have a hard time understanding the level of optimization that has been done. The academics typically do, in many cases, what I call titillating little studies. They say, oh, it works here, it works there. You have animal models, even some human data, that show that this really works. But they don't do the hard work of drug discovery. You have to look at solubility, you have to look at therapeutic window, you have to look at all the sort of assays for safety. The group that we're working with did a 5-year computerized structure-activity relationship optimization of this. Of course, today we'd call that AI, but it's been around for years. But and they optimized this drug in ways that were really unexpected, and that generated a whole new IP around the whole area that of this optimized class. So we think we're late preclinical stage. We want to do 1 or 2 more studies in the retina just to validate that this specific series of compounds does what all the other SEH inhibitors did. But there's actually been safety studies in human with some of these earlier generation drugs, and you can downregulate this enzyme to 0, and it still seems to be very safe.
So with that said, I want to invite over to the podium James Murray and David Esposito for RNL. Hi.
Okay.
Have a seat, have a mic.
And while we're waiting for James, I'm just going to put
James
in
is coming. Hi.
a pitch for the bullpen, because James and I actually connected earlier this week on the bullpen, and after doing so, realized we were going to be on the stage together, but only after doing so.
Great organization. That was the first time I'd participated.
Yeah, no,
Good to see you again.
It's entrepreneurs helping other entrepreneurs, and it's gotten quite a bit of momentum lately. Sorry.
So James, I think the first question is for you. So when evaluating a platform technology in ophthalmology as an investor, how do you assess potential scalability, long-term value, especially given the fact that this is a high-risk nature in a multi-asset company? What are key factors that make you comfortable to make an investment in such an asset?
Great question. So first, we love platform technologies, right? We think the eye is a great proving ground for emerging technologies. We actually invested in a San Diego startup called DTX, and their initial indication was ophthalmic, but ultimately it was a non-ophthalmic indication that led to them getting acquired, right? So we love the space, and I think first and foremost, what you need to think about when you're looking at a platform technology, is it truly a platform technology? Meaning, does that underlying technology and the advancement lead to efficient development of products, right? Or is it a product with multiple indications and applications, right? And I think the power of both of those is key. So once you answer that first question, it's platform technology, then the next question is, okay, well, what product, and you guys were touching on this, what product are we going to develop? So the number 2 question is, what product efficiently can we develop to de-risk and prove out this platform technology? And then finally, the third is, well, how are we going to monetize these different products? And how are we going to go to market and drive value? And what's going to appeal not just to potential partners, but also to other investors?
Yes. So I view this
We can hear you.
there we go. I view this platform as a little bit about the target, addressing the biology of these fats. When you have these degenerative diseases, and let's just lump them all together as degenerative diseases that are untreatable behind the blood-brain barrier, what do you see happening? You see complement activation, you see microglial activation, you see aggregated proteins and fats that get insoluble, things like drusen and amyloid and taus, or synucleins even. And all of those things are going on at the same time. You see lipid peroxidation, you see mitochondrial dysfunction. Which one causes which is not really known. But we think it's pretty nice as a platform to have a sort of a master switch kind of target that is pleiotropically downregulating all those bad things. We don't have to worry about which one causes which if we can look at them all, and maybe that's what's needed and why some of the single target approaches don't work as well as they should.
Does this causality or does this explanation makes your mind at ease? Does that convince you of anything?
We're getting there. We're getting there, right? But that's the hard work, is figuring out what you're going to address and how you're going to develop that phase 1 or so, and what indication you're initially going to pursue that's going to de-risk the asset and prove out your underlying thesis and the underlying technology.
Right.
I saw a stat that was the product and platform technology together extremely valuable, right? The pre-money median valuation on a product plus platform is $200 million. These are ballpark numbers. Product only is 130, and platform technology only without product is 60 million. So you see a really wide variation there. So you've really got to take that platform technology, that initial insight, and then get that first product out there. And I think some of the other comments during the previous discussion alluded to that as well.
Yeah, these are stark differences as far as valuations, absolutely. So David, you're looking at it from the other side. As a company that has successfully raised funds for a platform technology with multiple applications, how did you convince investors to invest in the broadened potential this has, and not just a one core asset?
No, thanks for the question. And James is one of our early investors, so I could also ask him on that question. But I think, in a lot of ways, it's pretty fundamental on what is the value of the scientific data across those potential, in our case, a small peptide that could impact a number of indications. What's the value of those indications? How complicated is the science to prove that out? And ultimately build a thoughtful plan to do that. In our case, it was the choices between an acute indication where we could prove it out pretty quickly versus chronic indications, and that became part of our overall investment thesis. Really prove out the science in the acute indication to then broaden into chronic indications. Just one aside to Tracy's discussion earlier, the Inflation Reduction Act and some governmental plans like that put some wrenches in the works where now you really think about indication sequencing more so for a small peptide around going big indications versus small. So you got to be fluid in the decision making, but I think for most of the groups here and as you work with investors, putting together that thoughtful investment thesis, I won't say it's basic, but it's fundamental. The team's got to work it out. In my journey of doing this for a little while, I've also taken 2 companies, controlled wind down, bankruptcy, however politely you wanted to say it. But it was really more around the inability for leadership teams and boards to coalesce around that ultimate strategy. Securing the funding is one thing, building the plan, but it gets very tempting to to what Bob just put out too, to want to go in a lot of different directions, and they all seem good, and the next scientific journal gets everybody excited. But you got to maintain a level of discipline at the board and at the executive team to just hum along in that base case plan. Otherwise, you can really get twisted.
And David, what are unique multi-asset strategic hurdles in managing this? So when you have a multi-asset program, how do you communicate this to investors with all these complexities, the potential values, the potential to simultaneously advance multiple assets versus prioritization?
It does get a little tricky, especially in our case with a lead compound that can hit a number of these, because you have some very practical parallel path clinical trial issues that can come up, whether it's AEs or new findings, all of those kind of things that can really trip you up. I do think it gets back to a pretty disciplined sequence of events around prioritizing indications to ultimately do that. I mean, there are opportunities around to be able to, and there have been investors that we've approached over time that do desire a little bit of shotgun start, because across a number of opportunities, because the other side that you're always pressured against is not just the great data, and it's not just your cash runway, it's the IP runway, and how you ultimately can get some of these to market to create the kind of value that investors want in the return. So a little about just comes back to fundamentally on building that operating plan and executing in a very disciplined manner, but also being prepared to pivot if you're running some parallel programs and one causes it. You know.
And James, how are you from the other side as an investor? How involved are you in this day-to-day decision making for the multi-asset?
In good cases, usually, periodically, maybe quarterly check-ins or so. But when things are going poorly, much more involved. Then they're reaching out, and it's some of what David was alluding to here, right? With the dysfunction and trying to focus and saying, "Oh, we need to do this, we need to do that," because I think that's the temptation with platform technologies, is that there's so much desire to solve all these kind of cool indications and go after this cool science, but you really need to focus. And the value of a platform technology is the ability to kind of leverage early data sets and to catapult that and to pivot and drive additional value if need be, right? So hopefully we're not very involved. We're certainly involved around the time of the investment and diligence and thinking through that with the company. But then, hopefully you have good leadership teams like O&L, and they just drive the company forward and can make those hard decisions, and they'll reach out if they want your input.
Yeah, absolutely.
James has called us to the late night principal's office every now and then to make sure we adjust the plan, but it's usually few and far between.
So I have a question for the panel. This area, looking at the fat disequilibrium in these tissues, is pretty bleeding edge stuff. I mean, we've had technologies that allow very high throughput and capable analysis of proteins and peptides and DNAs and RNAs for quite some time, and those areas have been, in some cases, mined quite extensively. And let's just use Alzheimer's as a case. We can now eliminate all the amyloids, all the taus, and people still don't get much better. What is your thought about one of this whole new areas where you're opening up that new technology area of saying, okay, we need to understand this enormously complex fat metabolome and how all these things interact of the types I was saying earlier in terms of different triggers for disease, versus something that's more known, something where you can take the next siRNA and make a really good platform play with all the right kind of bells and whistles it takes to make that work. How do you think about that dichotomy?
Well, I think, again, it comes...
No pressure.
None at all. Well, that's the temptation, right? Where there are many unknowns, but a huge unmet need to take the technology in that direction. But sometimes you have to take something that's a bit more understood and apply the technology to that pathway, right? And develop your data set. And then you can go after an area where there are unknowns. And you can also use For those other programs, use non-dilutive funding, right? The company I mentioned did a good job of doing that. You can also use early feasibility studies. Another company, I see Michael Rourke out there at Ravana, that is a sustained delivery technology, right? So multiple indications across different technologies and really utilizing that to make other drugs better. So sometimes you've just got to go after what you know, right? Or better understand, and then you can, go after kind of the bigger markets, the really high unmet need.
I may just add a few things. I think when you're venturing into new space like that, it always takes longer than you think to engage investors, to raise the kind of capital, to build out the team. It's probably exponentially longer when you're really breaking new ground. And so I think we all have to kind of put that in our plan that, one, it's going to take longer. But I do think your outreach to investors is much broader, and by virtue of trying to get some interest and excitement on that kind of platform. I won't say it's easy if there's a known pathway and you've got competitors, but people get it when you show up, and so you can move a little quicker in the process. But when you're really venturing into new space, you just gotta put the calendar out that's going to take a little bit longer, and I got to spread the outreach a whole lot wider to try to get some of that interest, because there'll be a few courageous few, but they're probably not on everybody's checklist, because a lot of those may not be ready to take the next step.
Fair enough. Any questions from the audience to our panel members? Okay, so I'm going to have to wake you up now. We're going to finish with the candy. Gentlemen, thank you so much. James, you're staying with me.
Okay.